Renee is going to tell us about the work that she does at the National Center for Genetic Resource Preservation. Please help me welcome Renee.
Welcome to the National Center for Genetic Resource Preservation. What are genetic resources? Genetic resources are the building blocks of life, such as the DNA of plants, animals, and microbes. For plants, this could include pollen, seeds, or buds. For animals, eggs, sperm, or embryos. And microbes, this includes yeast, fungus, and bacteria. Why would we want to preserve genetic resources? It is important to store genetic resources, not only for today, but for future generations. Some varieties may show resistance to insects, drought, or disease. Some may be new varieties that have multiple traits. Samples can be stored for conservation, such as rare and endangered species, or wild crop relatives, and revegetative species. Material is collected from around the world and sent to Fort Collins for long-term storage. In Fort Collins, at our facility, we test seed for viability and determine what is the best way to store samples. We store samples either we cryopreserve them in liquid nitrogen, or we store them in a cold vault. Seeds that are kept at -18 degrees Celsius, which is a freezer, are expected to be preserved for approximately 100 years. Samples that are stored in liquid nitrogen are expected to last 500 to 1,000 years. We haven't been doing this for 500 to 1,000 years, so we have been doing it for 100 years, and we do have seeds that's 100 years old that is still viable. So that's just a rough estimate. And then we put the seeds away for safekeeping. The National Center for Genetic Resource Preservation is part of the United States Department of Agriculture, Agricultural Research Services. It's really a bunch of letters. The mission of the National Center for Genetic Resource Preservation is to acquire, evaluate, preserve, and provide a national collection of genetic resources to secure the biological diversity that underpins a sustainable U.S. agricultural economy through diligent stewardship, research, and communication. E.O. Wilson had said, "We should preserve every scrap of biodiversity as priceless while we learn to use it and come to understand what it means to humanity." Early farmers and immigrants came to America, and with them they brought seeds and livestock from their home countries. They began their new lives in the New World, but with Old World agricultural stocks. The U.S. decided to preserve these valuable genetic resources. Officially, the U.S. began gene banking in the 1890s with the establishment of the first plant introduction, or PI, stations across the country. These introduction stations were used to evaluate plants with interesting traits, as well as develop long-term storage ideas. The first official sample was a cabbage from Siberia. In 1958, the National Seed Storage Lab was built to consolidate plant collections from not only the plant introduction stations across the U.S., as well as plant collections from private and publicly funded breeders into a single facility with state-of-the-art practices. Researchers at the National Seed Storage Lab began long-term studies to determine how water and temperature contribute to the shelf life of seeds and how these parameters had different effects on different species. The result of this research led to the bold, at the time, move in 1977 to convert seed storage vaults from refrigerators at 5 degrees Celsius to freezers at negative 18 degrees Celsius. Application of the modern field of cryobiology, just established in the 1950s, led to experiments to test whether seeds could be stored in liquid nitrogen. These experiments began in the 1977, but by the 1990s, liquid nitrogen storage was routine. Preservation of vegetative cuttings, as well as recalcitrant seeds, which are germplasm that requires water to survive, is now possible with cryopreservation facilities at the National Seed Storage Lab. Long-term storage could include not only the move from refrigerators to freezers, but could include storage in liquid nitrogen as well. In 1992, the National Seed Storage Laboratory was expanded, and the capacity to store germplasm increased tenfold. Some of the seeds that are stored there are corn, wheat, sorghum, soybean, cotton. Because it is part of the United States Department of Agriculture, we do focus on agriculturally viable samples. But not everything stores well as seeds. So we also store clonal samples, potatoes, strawberries, apple. In Fort Collins, we have almost 1,500 different genus of seed samples, with over 2,000 in the system, including all of the PI stations throughout the U.S. It's over 2,000 across the U.S. There are over 8,700 different species in Fort Collins, and 425,000 unique samples, with 583,000 in the U.S., plus an additional 300,000 samples, which would include botanic garden collections that we store for them, and an additional 5,000 vegetatively propagated. The knowledge about gene banking and preservation, as well as access to liquid nitrogen facility and high security at the renovated National Seed Storage Lab facility, made it an attractive place to establish the USDA's first animal gene bank. The National Animal Germplasm Program was initiated in 1999. The United States contains a broad array of livestock genetic resources that have been used by the livestock industry to improve production, efficiency, and increase food security. The National Animal Germplasm Program provides genetic security and increased understanding of these livestock populations for industry and research community use. The program interacts with international organizations in discussing policies concerning animal genetic diversity on a global scale and developing standardized methods for establishing and operating gene banks for livestock species. These are the animals we store: chicken, fish, pig, goat, sheep, turkey, beef and dairy, and bison. Again, we focus on agriculturally viable species. The preservation of plant-associated microbes became part of the center in 2009. The large state-of-the-art cryopreservation facility provides a secure backup of yeast, fungus, bacteria, and viruses, which represent a diverse and important component of U.S. agriculture. The National Seed Storage Lab's expanded mission to preserve more than just seed and more than just plant genetic resources prompted the designation of the facility as a center, hence the name change to the National Center for Genetic Resource Preservation in 2002. The mission of the National Center for Genetic Resource Preservation, again, is to acquire, evaluate, preserve, and provide a national collection of genetic resources to secure the biological diversity that underpins a sustainable U.S. agriculture economy through diligent stewardship, research, and communication. Are there any questions? Okay.